I want to discuss the effects of hormones while you and I were separately in utero, and the effects that that had on who we are, who we select as mates, so mate choice, sexual preference, and all other aspects of what you would call sexual development. Now, this is something that's gotten a lot of popular press, and it has to do with how exposure to androgens in particular, while we were in utero, impacted whether or not people report as homosexual, heterosexual, identify as male or female. There was a researcher who's still going now. His name is Dennis McFadden. I believe he was at UT Austin back then. And he was studying the auditory system. And people would come into his clinic and he would, or his laboratory, and he would look at hearing, and he would explore different aspects of what they call the psychophysics of hearing and understanding hearing thresholds and frequency thresholds. And he made several observations. And those observations were that young males tended to have what are called autoacoustic emissions more often than young females did. Autoacoustic emissions, as the name suggests, are the ears actually making sounds. Now, these sounds have to be picked up by a special apparatus because they can hear into that frequency, but it turns out that your ears don't just take sound waves and convert them into these things that this thing we call hearing, but they also, in some cases, make sounds. So your ears are making sounds. Strange, right? So it turns out that there's a sex difference in autoacoustic emissions. Turns out also that people that self-report as lesbians, they also have autoacoustic emissions significantly more than females that don't self-report as lesbian. And Dennis noticed this and published this, and it was an important discovery because it was one of the first discoveries that that pointed to the fact that there are sex differences in biology that are independent of sex. I mean, this is hearing and autoacoustic emissions. And just to really illustrate what the former problem was and why this study was so important, a lot of people had explored, for instance, whether or not homosexuals had lower testosterone, for instance, in males. And actually, the result often was the opposite, that gay men or men that self-report as gay often had much higher testosterone. And those studies then became controversial because people said, well, sexual behavior can relate to testosterone, etc. And so it became very controversial. And then there were some studies that attempted to look at the equivalent phenomenon in people that self-report as lesbian or self-report as heterosexual. And so it became very complicated. But this was an identification of a phenomenon, autoacoustic emissions, that was independent of anything that had to do with sexual or even social behavior. 1998 rolls around, and I'm a graduate student at UC Berkeley, and a guy by the name of Mark Breedlove, kind of an ironic name given that he worked and still works on sexual dimorphism in the brain and in the spinal cord and nervous system. And Mark, who's a phenomenal scientist, comes running down the hall. I'll never forget this. And he said, "Give me your hands." So he said, "Give me your hands." And he pulls out a ruler and he starts measuring my fingers. And he takes down a couple measurements and then he goes away. And I'm like, what was that? Well, I was in a course that Mark was teaching at that point. And soon after, we did a study that Mark directed, exploring the finger length ratios, and I'll explain what those are, of males and females and people that self-reported as homosexual or heterosexual. So let's just get to the basic, what we'll call sex differences first. These are averages. I want to point out, anytime you get into this kind of topic, people are, assume it's causal, but it's not causal. These are averages that I'm about to report. It is the case that the ratio of what's called the D2 to D4 digits. So the D2 is your index finger. So your thumb is D1, then D2 would be your index finger that you would point with. Middle finger is D3, which you whatever with. And then D4 is the so-called ring finger. Okay, and D5 is the pinky. It is the case that the D2 to D4 ratio is greater in self-reported females than it is in males. What does that mean? It means that digit D2 and D4 are more similar in length in females than in males, and the effect is particularly, pronounced on the right hand, although not always, okay? And it does not have to do with handedness. This D2 to D4 difference has to be measured correctly. You can't just look at somebody's hands and say, oh, their ring finger and index finger are very similar, and therefore they are female or they were exposed to very little testosterone in utero. You can't look at somebody and see that their index finger is much shorter than their ring finger and say, oh, they must have been exposed to a lot of androgen. You have to actually measure it, and you have to measure it correctly. You have to measure it from the base of the finger where there's that first crease all the way to the tip, past the, you can't include the fingernails if you're growing fingernails. It would be logical here, folks. So you can't normally see it from the back of the hand, although I don't know if this will show up here, but if you look at the back of the hand, sometimes you can see it. In my case, for instance, let me see if I can do this. So my D4 is a little bit longer than my D2. In some people, it's more pronounced, and that's on my right hand. On the other hand, the difference actually is far less pronounced. It's a little bit pronounced there, but not so much, okay? So that's sort of the typical ratio that you would see. Turns out that in mice and in humans, the more androgen that you are exposed to in utero, the smaller the D4, D2 ratio, meaning that the ring finger tends to be slightly longer than the pointer finger. And in females, because they're exposed to less androgen in utero, typically, then those fingers tend to be more equal in length. And these are subtle differences, and these are averages. I invite you to look up the paper. This was published in Nature in 2000, and it's been replicated 6 times. Now, here's where it gets even more interesting and potentially precarious, so we're going to step cautiously here. If you look at the finger length ratios of men that self-report as homosexual, they have either the typical male pattern of D2 to D4 ratio or a hyper-masculinized D4 to D2 ratio. Now, this can't be something that's established or modified by behavior. This has to be something that was established in utero, and in fact, it's present at birth. Okay, so it completely divorces the interactions between hormones and behavior. And that's an important theme that we've been talking about, and we're going to talk about even more next episode, is that hormones impact behavior, but behavior also impacts hormones. But this is a case of hormones impacting what really should be considered a primary sexual characteristic, because it doesn't show up in puberty, it shows up before puberty, it's actually established in utero. And in people that self-reported lesbians, and I remember going out there and collecting these data with the collaborators on this work. Again, I wasn't the main driver on the work, but I participated in some of the analysis. People that self-report as lesbians also tend to have a smaller D2 to D4 ratio. So this is consistent with the autoacoustic emission study that Dennis McFadden had published, and it points to the fact that early exposure to androgens may have an impact not just on androgenization of the body plan, but also separately on sexual preference. Now, this raises all sorts of interesting questions about biological basis of sexual preference. I'll tell you about another study, a guy named Simon LeVay, who was at UCLA, who trained under Hubel and Wiesel. If any of you remember early episodes on plasticity, David Hubel and Torsten Wiesel, my scientific great-grandparents, won the Nobel Prize for discovery of critical periods for brain plasticity. They define some of the most important aspects of how we see and brain plasticity. Simon LeVay trained with them, and then Simon went on to discover that in the brains of people that self-report homosexual, there is a brain difference. And the brain difference is in an area called the interstitial nucleus of the anterior hypothalamus. So it's the INAH. And so there are published reports, that was published in Science. The other work I referred to was published in Nature and then replicated no fewer than 6 times, and the McFadden results that point to strong biological correlates of mate choice, of sexual preference. And these tie directly to things like androgenization or estrogenization, meaning we could call it maleness or femaleness, but that's sort of tricky territory because of the way that we described the huge range in which sex can be defined earlier. So if you want to measure D2, D4 ratio, you're welcome to, but you also have to understand that it's not predictive of anything, right? It's just a window into the possible androgen exposure that you had early in life. There are plenty of men who report themselves as heterosexual who are out there who have similar or have D2, D4 ratios to females. And there are plenty of females whose index fingers are shorter than their ring fingers, and they're perfectly happy, or they say they're perfectly happy, and we are inclined to believe them, being heterosexual. So there's variation. In fact, Mark tells a really good joke. If you want to know whether or not somebody is homosexual or heterosexual, simply look at their hands, look at their D2, D4 ratio, and guess heterosexual, and you'll be right 96% of the time. Because 96% of the time, people report themselves as heterosexual. On average, those numbers might be changing. So the joke really is a joke on science, because that falls within the realm of statistical significance, and yet it really illustrates the fact that none of this is causal. But it's nonetheless very interesting because it means that hormones are organizing the brain early in development in ways that can potentially impact same or opposite sex partner choice later in life. Now, of course, there are other things that can impact opposite sex or same-sex partner choice later in life. The study did not look at people who reported bisexual. There hasn't been a lot of studies on that yet. One thing that's very interesting, for which there's some good scientific data, but there's also some controversy, is that it appears that the probability of a male human self-reporting as homosexual increases with the number of older brothers that he has. Now, that doesn't mean if you have an older brother, or even if you have 10 older brothers, that you are sure to self-report as homosexual. But statistically, it becomes more likely that somebody will with each successive older brother that they have. And the idea that's starting to emerge in the developmental neuroendocrinology landscape is that there's a record within the mother of how many male fetuses she's carried, because male fetuses are secreting certain things, dihydrotestosterone, other things, that can feed back onto the genome. So these could be epigenomic effects, or onto the placenta itself, so that there's a higher probability in subsequent pregnancies that offspring will self-report as homosexual. So it's a fascinating area of biology. And as you've noticed today, none of this deals with the current controversies around gender and how many genders and sex, etc. That's a separate conversation that is, by definition, grounded in the kind of concepts we've been talking about today, and needs to take place taking into consideration all of the aspects of sex and the effects of hormones, both on the body, on the brain. We didn't talk a lot about spinal cord, but we will in the next episode, but we can just say on the brain and the periphery.